So, like I said before, with temperature and pH, the factors are going to be directly tied to whether they can denature an enzyme or not. Okay. So below we have with temperature and pH, there is an optimal range that the enzymes will function at, meaning that they want to be at that temperature and they want to be at that optimal pH range. Anytime for enzymes, they go beyond or below the optimal range, there is a chance it can denature. For temperature, if it goes above, it will definitely denature. And what does denaturing an enzyme mean? Can I get Amalinda? So an enzyme has denatured. What does that actually mean? So what is, if I wanted you to describe it in an exam, how would you describe that? So first you mentioned the enzyme has denatured. That's your first point. What is the other 2 points that you need to mention? So that's why it denatured. That's part of the first sentence. So the other 2 sentences, you need to mention is first, conformational change at the active sites. So remember that enzymes function by letting substrate bind to the active site. Because due to the heat, the structure of the enzymes are broken down, so the conformational change at the active site occurs. As a result, the enzyme loses its function to catalyze any chemical reactions. So anytime we go above the optimal temperature range, that's the 3 things that you need to mention. First, enzymes are denatured. Second, conformational change at the active site. Third, due to that, they lose function. So you can just combine the second and the third one together. So by saying that there is a conformational change at the active site, it loses its function. You can get the best outcome. All right. So that is when it's above the optimal. But however, when it goes below, what happens is that the number of collisions that the enzyme and the substrate experience are much lesser due to the lower amount of kinetic energy. And essentially, this will slow down the reaction more and more until it gets to a point where the enzymes are literally frozen. Okay. What is one thing that's different about going below the range versus going above the range in terms of enzyme function? Okay. So when we go down, enzymes lose their function too. And when we go above, enzymes lose their function too. But what is the difference between them? Can I get Abi? Exactly, right? So essentially, when we go lower, there is a loss of function, but what's happening is that the enzymes are basically just getting slowed down to a point where they are frozen. So essentially, they can regain their function back if you bring them back to an optimal temperature, because essentially the function of a protein is a function of an enzyme or a protein is tied to its structure, how it's shaped. And when you lower the temperature, you're not changing the shape. You're just lowering its kinetic energy. But when you're going above the optimal, you are breaking down the shape of a protein or an enzyme, making it non-functional, and that is a permanent change. You can't go back once you change the shape of a protein. You can only get function if you have frozen it and you are unfreezing it. Okay, that is for temperature. For pH, the case is slightly different in that it has the optimal pH range, and anytime you go above or beyond, it will automatically denature. Because pH is not the same as temperature. Anytime it's too acidic, anytime it's too basic, it will denature. So how do we determine what the optimal pH range for any biochemical reaction is depending on where the biochemical reaction is occurring. Remember that the 3 stages of aerobic cellular respiration happens at different places, right? The first one happens at the cytoplasm, the second one happens at the mitochondrial matrix, and the third one happens at the cristea. Okay? Each of them has their own optimal pH range. So, for example, the cytoplasm is sitting about 7.2, whereas the mitochondrial matrix is at a 7.8. So the enzymes that are functioning at those different locations have to be suited to those pH ranges. And if they are placed in any other environment, they will immediately lose their function and denature and then lose their function. Okay, any questions about this? Okay, both of them are talking about enzymes and how enzymes are going to be denatured if it's above the, if it's not within the optimal range. That's the key point here. And this is the same for photosynthesis as well, right? Exactly the same. Photosynthesis, there's the optimal temperature, there's the optimal pH. If it goes beyond those optimal ranges, then it loses its function. Okay. So one thing to note about this is that with, let me just move this out of the way. Select. Okay, so the unfortunate reality with temperature and cellular respiration, if you guys haven't realized, is that the more you use your energy, the more you build body heat, right? Your body naturally gets hotter the more you do physical activities. And that is a vicious cycle because with temperature, like I said before, it can influence the activity of the enzymes. And when I say that if it goes above the range, it starts to lose function. The range is actually a very small range, right? Between 37 to 38 degrees Celsius, that's where most enzymes are most efficient, especially within your body. And anytime you go beyond that, they start to denature and lose function. So the problem is, if we are undertaking too many physical activities, we are going to increase our body temperature, right? And as a result, we will start to break down the enzymes that are working for cellular respiration. And basically, any enzymes in our body are going to be affected because body temperature increases above the optimal range, and then enzymes start to denature. So, what do we do to fix this situation? What is the one thing we do to reduce our body heat from going too high? Any ideas? Something very simple. Everyone does it. Sweating, right? Sweating is our body's natural system to cool our body in such a way that when you sweat, the act of sweating itself is not what's causing you to cool down. The act of the sweat evaporating off you, that is what's taking the heat away. That is what's keeping your body at a lower temperature such that it will not allow the enzymes to denature. Okay, there are situations where this is not even enough, but we don't have to worry about that. That is when you get something known as heat stroke, where your body just continually increases in its temperature to the point where your body just can't survive anymore. All right. So next factor we're looking at glucose availability. Right. This is the substrate involved with the enzymes. The first requirement for any type of cellular respiration to occur, whether it's aerobic or anaerobic, you need glucose as your reactant if you want anything to happen, because glucose is the input for glycolysis, which is the common stage between the aerobic and anaerobic. By increasing the glucose concentration, as always, we will increase the rate of respiration. And as always, there is a point of saturation where it's basically saturated all the enzymes with all the glucose it has, and any increase in the glucose won't change the rate of reaction. Okay, remember, it's the same with photosynthesis and it's the same with any enzyme related topics. There's always going to be a point of saturation where all the enzymes have all the glucose they need. So any more glucose you give won't do any different, won't change anything, right? So at that maximum level, the respiration rate will plateau, as we've looked at in the past. And this is because the enzymes involved in respiration have reached their saturation point, in which case another factor has become the limiting factor, right? So you could say that enzyme availability is the best limiting factor here, because there isn't any enzymes to take up any more glucose, any extra glucose. So the rate of respiration has plateaued and will stay consistent. All right. Any questions about this? Okay. You just need to understand what is the saturation point and the limiting factor once, and you can apply it in a lot of different contexts. Because anytime enzymes are involved, there will be a saturation point, there will be limiting factors. Okay. So you can always just remember those key ideas and reapply them over and over. Next is oxygen availability. Obviously, this factor plays into whether the cell undertakes aerobic respiration or anaerobic respiration. Essentially, if there is oxygen available, it will do aerobic. If there isn't any oxygen available or a very low amount of oxygen available, it will do anaerobic respiration. So we also know energy production between aerobic and anaerobic is very different. Aerobic gives you 32 ATP, whereas anaerobic gives you 2 ATP only. So essentially, the cell itself wants to do aerobic respiration, but only if there's an inadequate amount of oxygen will it undertake anaerobic respiration. All right. So essentially, if you give it enough oxygen, it will start to undertake aerobic respiration. But as always with enzymes, there's a point of saturation where the enzymes literally can't take up any more oxygen. So enzyme availability will become your limiting factor at the saturation point, causing it to plateau and have a consistent rate of aerobic respiration. If you go to a very low scale here, where it shows a rate of aerobic respiration very low, what is happening instead is that more anaerobic respiration is happening. So if you had another graph showing anaerobic respiration, it would be at a very high rate at the same point. Any questions about this? Zoe, it seems you have a... Right? Essentially, we take up oxygen. If there isn't any oxygen, your body starts doing anaerobic. So this graph here is showing rate of aerobic respiration. So obviously, at a low oxygen concentration, it's a very low amount of aerobic. But if we had another graph that is showing rate of anaerobic respiration, at a low oxygen concentration, it would probably be at this high point and then go lower and lower as... So they would probably have a inverse relationship. So for this, it goes like this. For anaerobic, it will just sink to the bottom as soon as there's a decent amount of oxygen concentration. Any questions? Moving on. And in enzyme inhibition, this one, like this, we've talked about a bunch with enzymes. Enzymes can be inhibited so such that they don't actually catalyze any biochemical reactions. There's two types of inhibition that can occur. There's one that's competitive, the other one is non-competitive. Competitive is called such because it's competing to bind to the active site of the enzyme, right? The inhibitor molecule wants to bind to the active site of the enzyme such that it prevents any substrate to bind to it afterwards, right? So this is what we're looking at for competitive, where it binds to the active site, and because it's bound to the active site, it will not allow any substrate to bind to it anymore, basically preventing the catalysis from happening. Non-competitive inhibition is non-competitive because it's not competing to bind to the active site. The inhibitor molecule just needs to bind to the allosteric site around the enzyme, and it will cause a conformational change at the active site. And again, as we mentioned with the denaturing part, whenever we say conformational change at the active site, that will prevent the enzyme from carrying out its intended function. So when we're thinking about competitive versus non-competitive, we also need to consider whether it's a reversible inhibition or it's a permanent inhibition. So when it comes to non-competitive inhibition, more often than not, it is permanent, where the inhibitor molecule is not forced to unbind or will not unbind very easily. With competitive inhibition, the inhibitor molecule can actually be forced to unbind from the enzyme under one condition, where there is a high concentration of substrate present in the environment. If there's a high concentration of substrate, then the inhibitor molecule will naturally unbind from the enzyme and allow the substrate to bind to it. Okay. However, there are some cases where the inhibitor molecule has very strong bonds with the enzyme as well. That is when we call it as permanent inhibition, or what's the proper word for it? So it is non-reversible inhibition. So if the inhibitor molecule can be forced out, we call that as reversible inhibition. If it's sticking to the enzyme and no matter how much substrate is present, it never unbinds, that is non-reversible inhibition. Okay, so we'll talk about that in a bit. So essentially, let's look at this graph right here, right? So this graph will explain what we just looked at. So this graph, the y-axis is the rate of reaction, the x-axis is substrate concentration. And remember what I said about substrate concentration and competitive reversible inhibitor, right? So the green graph here, that shows the rate of reaction, the rate of reaction under a normal situation where there's no inhibition. So essentially it rises and rises until it reaches a saturation point where enzyme availability becomes the limiting factor. So it plateaus and stays consistent. Okay. Now the red one, we see this as non-competitive or irreversible inhibition. This means that the inhibitor molecule has bound to the enzyme and won't let go of the enzyme no matter what, because the bonds that they have is that strong. So essentially, if you started out with 100 enzymes and you got the uninhibited graph, with this red one, out of the 100 enzymes that were available, 50 have been deactivated. So essentially, because 50 have been permanently deactivated, you can see that the rate of reaction is permanently stunted. It won't ever go above this line because out of the 100 that could have been able to increase the rate of reaction, only 50 were available. So you've basically reduced the rate of reaction by half. Okay, any questions about this? Okay, that's why even the trend is exactly the same because everything else will happen exactly the same, right? The rate of reaction will keep increasing until it reaches a saturation point where enzyme availability becomes a limiting factor, causing the rate of reaction to plateau and stay consistent. So that is irreversible inhibitor. Now let's look at reversible inhibition, usually with competitive reversible inhibition. So essentially, the rate of reaction rises and rises until it reaches a point where the substrate concentration is overtaking the inhibitor molecule concentration. So once that happens, any enzymes with the inhibitor molecule bound to it will suddenly will become separate, where the enzymes will be free to allow substrate to bind to them because of the higher concentration of substrate. So this trend is slowly changing as you increase the substrate concentration because that's how you reverse a reversible inhibition. Okay, you increase the substrate concentration surrounding the enzyme, and naturally the enzyme, the inhibitor molecule will unbind from the enzyme. So yeah, so you can see the picture here, right? So with this with this blue graph, when it's increasing in substrate concentration, whatever inhibitor molecule is bound to the enzyme here will naturally unbind, allowing the substrate to bind to it. That is why after a certain point in the substrate concentration, it naturally returns back to the original rate of reaction, the maximum rate of reaction, because at this point, all the inhibitor molecules have unbound from the enzyme, allowing some substrate to bind to it. And again, what naturally happens where it reaches a saturation point and enzyme availability becomes a limiting factor and the rate of reaction plateaus. Any questions about this? Okay. Irreversible inhibition because it will never let go of the enzyme. Okay, reversible inhibition because the inhibitor molecule can potentially let go of the enzyme if it is present in a high substrate concentration. All good? Okay. So one thing to note is that in cellular respiration, it's also something known as feedback inhibition that occurs where the inhibitor molecule is also happens to be the end product of the entire biochemical reaction, biochemical pathway, where if we started cellular respiration here, what is the final product we want to get? Can I get Yaput? Okay, this is the start of cellular respiration. What is the final end product? what do we want to get out of cellular respiration? What is the final goal? We want to break down glucose to get what? There's a name for it, right? The energy transfer molecule, the coenzyme. What's it called? It's a A with a T with a P. What does it stand for? ATP. There you go. All right, so the end product is ATP for cellular respiration. That end product actually acts as an inhibitor molecule for the first few stages of cellular respiration. Why is this the case, right? Imagine your body is producing a lot of energy because your body is an energy deficit. Basically, you need about 8,000 calories to survive every day, right? Wait, no, that's not right. Kilojoules of calories. So you need a certain amount of energy to survive every day, but your body doesn't need more energy than it actually. Your body doesn't need to break down more energy than it actually needs. In fact, it doesn't want to do that. It wants to store any extra potential glucose to be used later when it's actually needed. That's why, fats, adipose tissue, all that happens because your body is basically determining you don't need to make this much energy. You can keep this glucose for later when we actually need it, right? So what happens is that once your body has produced enough ATP, that ATP acts to inhibit cellular respiration. Okay, by inhibiting cellular respiration, it in turn reduces ATP production because at this point there's so much ATP, it doesn't need to make any more, so it inhibits itself from undertaking cellular respiration. That is what it's called to be a end product inhibition. Is when the uh, so, like I said, that is what an end product inhibition is referring to. The end product is inhibiting the cellular respiration process itself. And this is actually a common thing that you can see all throughout biology, where the end product ends up inhibiting the process. You should probably go to study space, right? Is that how it works? Yeah, just come in. We're basically done. Well, how are we basically done in the first 30 minutes? Right, we'll do revision for the rest. All right, like I said, end product inhibition is when the end product of the entire biochemical pathway acts to inhibit the biochemical pathway. So it could inhibit it in separate stages. You don't really need to know which stage, but all you need to know is that the ATP molecule produced at the end acts to inhibit the process of cellular respiration if there's a high enough concentration. And this can be the case for photosynthesis as well, where an end product the end product of a certain chemical reaction acts to inhibit something in the earlier stages of photosynthesis. Okay, this is something very common in biology, just so that it acts to prevent an overproduction of a certain molecule, right? Even in genetics, it's present where the protein made from a gene can act to inhibit the expression of that gene so that you don't make too much of that protein as well. So end product inhibition is a very common and a very important step to ensure you don't waste your resources. You don't make something you don't need, and you don't make too much of whatever you need. Any questions here? All right. So far we looked at cellular respiration, looked at photosynthesis. Now that you guys understand the processes involved, what's going in and what's coming out, let's do a very simple summary and make sure you guys remember something very important. So with cellular respiration, let's start with photosynthesis. That's where the energy starts, right? So light energy from the sun comes into plants. What pigment is responsible for absorbing that light energy and storing it? I get. All right. So what pigment in plants is responsible for absorbing light energy and storing it to be used later in the chemical reactions? Yeah, chlorophyll, right? That's a green pigment. That's why a lot of the plants you see around you are green. It's the chlorophyll within them That's giving them that green color. Okay, so light energy comes in, chlorophyll absorbs that light energy and uses it for photosynthesis. So there's two stages that occur in photosynthesis. Can I get Ayman? What are the two stages involved in photosynthesis? The names very much explain what. Actually, yeah. Can you tell me? I don't want to give too many hints. Any ideas? No? Can I get Yasmin? Okay, what are the two stages involved in photosynthesis? Light dependent, light independent. Okay, the names are that easy to remember and they explain the stages, right? Light dependent, what does it need? Light. Light independent, what does it not need? Light. Okay, it says in the name light dependent, light independent. So you just have to remember that. After those two stages, what is the final product of photosynthesis, Gavin? Yes, make sure we look it up because that's going to be very much part of your SAC. Okay, that's actually one of the main focuses of your SAC is photosynthesis. In fact, out of the three experiments we're doing, two is about photosynthesis, one is about cellular respiration. Okay, I'm going to be releasing the the practical document by today and so that you guys can have a look. And you guys are gonna get about 20 to 30 minutes in tomorrow's lesson to fill it out. In the same way how we did it with your previous SAC as well. You'll get 20 minutes to fill it out, and then I'll give it to you back during the experiments for you to fill it out further. Okay, so essentially, photosynthesis has two stages to produce the final product of glucose. Okay, basically, what is photosynthesis doing is taking up carbon dioxide and H2O. It's taking light energy from the sun, undertaking photosynthesis to make glucose out of these reactants. So the reactants are CO2 and H2O. Light energy is not a reactant, it is a requirement. Okay, do not ever confuse that. Light energy is not a reactant of the reaction, it is a requirement. There's a difference such that it's not a molecule that you give or anything, it is something that needs to be present for it to undertake cellular respiration as well. Cellular respiration, photosynthesis at all. Okay, so with CO2 and H2O combined with light energy being present, the plant cells, the chloroplasts are able to undertake photosynthesis to produce glucose. This process takes up carbon from the atmosphere. Okay, so in the carbon cycle, if we looked at what is actually absorbing carbon from the atmosphere, plants are the main contributor to that. Through photosynthesis, where they take up carbon from carbon dioxide and convert it into glucose. Either they convert it into glucose or they store it within themselves. That's why you hear a lot of people say that, oh, forests are carbon sinks because they absorb so much carbon. And essentially, by cutting down forests, we are taking all the stored carbon, releasing it back into the atmosphere. And why is that a problem? You can kind of see why that's a problem already, so I don't need to explain. Okay, another byproduct of the photosynthesis reaction is oxygen, which is imperative for cellular respiration, right? So what goes in? CO2, H2O, light energy. What comes out? Glucose and oxygen, and also some H2O. Okay, and then that will be used for cellular respiration, right? The oxygen release and the glucose release is taken up for cellular respiration. And what are the two forms of cellular respiration? So this is basically revising what we revised at the start. Sorry, since you missed most of the lesson. Okay, aerobic and anaerobic. Which one produces more energy? Aerobic produces more energy, right? So essentially, oxygen and glucose is taken up into the mitochondria. So all three stages of aerobic cellular respiration done in the mitochondria. Okay, which stage is done outside of the mitochondria? Glycolysis, the first stage, right? And then the other two stages are done inside the mitochondria, where it produces heat energy and ATP, and also releases the byproduct of CO2 and H2O. If you ever have trouble remembering what goes in and what comes out, use cellular respiration as your point of reference, because you basically are a good model for you to understand what is a byproduct, because you are constantly releasing those byproducts, right? CO2 and H2O is constantly coming out of your breath because of this reaction, right? Your body is constantly making energy through cellular respiration, and that's why you are constantly taking in oxygen and releasing carbon dioxide and H2O through your breath. And that is taken up by plants. So you can immediately assume whatever you're releasing through your breath is what the plants need to take in, including light energy from the sun, to do photosynthesis for glucose. So you don't have to necessarily remember all the reactants and all the products. You just have to use one point of reference to be able to find them all out. All right. So yeah, so one thing I forgot to mention. So where plants take up the carbon in the atmosphere to make into glucose, basically storing it, cellular respiration basically releases that stored carbon in the form of carbon dioxide. But we also have a bunch of different things that we do that release the carbon dioxide that is stored in plants, such as burning fossil fuels and so on. In fact, that's what we're going to look at in your next chapter. The next topic is how we release some of the carbon that is stored in plants in the most horrible ways. Right? So these are the different graphs associated with each factor. For factor 1 and factor 2, obviously, it's showing temperature and pH, the optimal range. Anytime you go above or below the optimal range for pH, the enzyme will denature. For temperature, if you go below the optimal range, you will lower the kinetic energy, meaning you will lower the collision between the enzyme and the substrate, such that eventually the enzymes will all get frozen. Okay, the enzymes can regain their function if you just increase the temperature back again, because essentially the shape of the enzyme has not changed when you reduce the temperature. All that's happening is that they're getting slower and slower until they're frozen in place. So they can still become functional if you increase the temperature. However, if you go beyond the range to a higher temperature, the enzymes are starting to denature, where they will start to have conformational changes at the active sites, becoming non-functional. And that is a permanent change. Same thing with pH. If you go above, you go below, it becomes too acidic, too basic, the enzymes denature, lose their function, and this is a permanent change. Anytime enzymes denature, it is a permanent change because they lose their structure. And as we've looked at with proteins, anytime they lose their structure even a bit, they can become non-functional. With glucose concentration and oxygen concentration, it's the same, where there will be an increase in the rate of reaction until it reaches a saturation point, at which case it will start to plateau and become consistent, where the saturation point is caused by another limiting factor, right? Any questions? All right. Fun facts. So we've looked at oxygen availability and how that can trigger aerobic versus anaerobic, but some people have actually evolved to try to combat this. For example, there are people who live in a very high altitude. For example, sorry, not for example, a city in Peru is living at an altitude that's so high that the oxygen content in the air is very, very low compared to somewhere at ground level here. So how do the people there make sure that they're constantly not doing anaerobic respiration instead of aerobic? Is that their bodies have essentially evolved countermeasures to try to increase the oxygen extraction from the air. How do you extract more oxygen from the air when there's less oxygen in the air to begin with? Is by increasing the amount of red blood cells in your body. So think of red blood cells in your body as oxygen miners, right? Anytime you breathe air, you're breathing air mixture of different gases, right? You're breathing in nitrogen, you're breathing in oxygen, you're breathing in carbon dioxide, you're breathing in a bunch of different gases, and they all come with a fixed concentration, such that oxygen is only 21% approximately of the air that you breathe. So how do you manage to extract the 21% and only the 21% that you want? Is that your red blood cells specifically target the oxygen in the atmosphere. Sorry, when you breathe it in, they go into your lungs, and the red blood cells will specifically target oxygen to bind to them, such that they bring it to the cells that need those oxygen molecules. So by having more red blood cells, your body is essentially trying to extract oxygen harder from the air. So when is that necessary? Is when there is less oxygen in the air to begin with. So imagine there's like 15% instead of 20%. Your body needs to work harder to get it out of that 15%. So your body needs more red blood cells. Okay. If you have a condition where you have excess oxygen, then your body is going to be not needing that much red blood cells. Another condition where it's also is important is when your body can maximize the oxygen that you are taking in. Where is this relevant? Is for people who are going for long dives. So there are people who I've heard, not proven yet, I need to get more evidence about it, who can actually hold their breath up to 16 minutes when they want to go diving for hunting fish, because their body basically can absorb so much oxygen into their blood, and that can be used for a very long time compared to regular people, right? So there are different adaptations and different adaptations your body can make such that you become more resistant to these kind of conditions, right? Even if there's no less oxygen, how can you survive? What are the adaptations you can get? If you have a limited supply, how can you make the most out of it? There's also adaptations for that. Okay. Essentially, what biology can do is very limited. It's not limited to simple factors like this. Adaptations and evolution can actually give way to a lot of different things. For example, at first, oxygen was actually toxic to a lot of living things. So at the earlier stages of life, I'm going out of topic here, so just you want to listen, you can listen. At the earlier stages of Earth development, there wasn't a lot of oxygen present. In fact, the atmosphere was carbon dioxide rich. When the first photosynthesizing cell emerged, that's when oxygen started to be produced. And when that one cell started to mass produce and was all over the world, suddenly the atmosphere was getting filled with oxygen. So in the same way, if our current air got filled with carbon dioxide, we would get suffocated. Whatever was living back then got suffocated due to the excess amount of oxygen. Only after that, evolution occurred and living things that could actually use oxygen emerged. Before that, that wasn't the case. So through changes, evolution can actually keep up with those kind of changes, such that even in caves where there isn't any oxygen or carbon dioxide, there are cells that can actually break down different gases, right? Sulfur dioxide, for example, is a gas that certain microorganisms can break down to gain energy as well. So biology is very much not limited by whatever boundaries that you think there are. See, dinosaurs, that's when I say early development, I mean like billions of years ago, not millions. Dinosaurs was about 670 million. This is more about
670 million years.
yeah, that's when the dinosaurs went extinct. When the asteroid hit the what we can do archaeology. We can measure soil, we can see what we can find. So the thing about soil is that whatever happened at that time, the soil will create a deposit of it, right? If there was an excess amount of lava, there will be a soil deposit of that. And over many years, that soil deposit gets buried and buried. So essentially, if you could see the different layers of soil, you can kind of study what might have happened. And that's what we've done. That's what we've done to try to analyze what's happened in the past. Theoretical supported by a lot of evidence. See, everything in science is essentially starts out as an idea. We use evidence to support that idea, so much so that it becomes a theory. So you guys hear the word theory and you think, oh, that's actually something unproven. In science, that's the exact opposite. When we give the word theory to a certain idea, the theory of natural selection, the theory of uh, genetics, we are basically saying this is an idea that has countless studies that are supporting it and very little saying otherwise. So if you want to disprove it, you need to present an equally substantial amount of evidence. And that can happen as well. Science is tentative. Science uses empirical data. What does it mean for it to be tentative? It means that it can change depending on the studies, depending on the evidence that we have. Science is not fixed, right? In fact, if you think about uh, the structure of an atom, right, that you guys have learned when you were kids, that structure has changed so much over the last 100 years because more and more evidence was obtained about what it actually looks like or what it's actually structured like. So we change our understanding as such. But again, science is driven by evidence. If we have evidence, then we can make a claim. If we don't have evidence, then we're just having a hypothesis that we need to prove using an investigation. Okay. Again, this is really off topic from what we were looking at. All right, so yeah, so can I get Mary? Okay. Matthew investigated how changes in environmental temperature affected oxygen and carbon dioxide levels in the air around the cockroach. Okay, so basically, how does temperature affect how much oxygen or carbon dioxide is around the cockroach? What is the cellular process that they are investigating here? What can impact on the oxygen and CO2 levels around a living thing? But what changes it? So if I, like, let's say if I started to do an investigation of Earth and I said what could change the oxygen and CO2 levels in this room right now? What cellular process would change it? There's only one that we've learned so far that actually uses oxygen, releases CO2. A little something we've been learning for the past few weeks. Sorry? What's the full expression? What's the full what's the there you go. Yeah. Don't overthink it, guys. If you think that's the answer, that is the answer, right? It is cellular respiration. Or you could be specific and say aerobic because there is oxygen present. So it'll probably be aerobic cellular respiration. But essentially, it is cellular respiration. So just consider the factors, right? What is present? Oxygen and CO2. If they are measuring oxygen and CO2 levels, there is only one thing they care about, which is cellular respiration. Because what is needed for cellular respiration? Oxygen. What is released as a result? CO2. And they are looking at whether temperature is affecting it. So it's essentially about cellular respiration. Okay, what is the dependent variable? What is the independent variable? Again, this is very much related to your sets. Make sure you guys get good at identifying your variables. Can I get Abi? What is the dependent variable here? Okay, what is a dependent variable? Explain that first.
The variable that doesn't change.
That doesn't change. That does change. Yeah, that's the point. You need to know. Even if I confuse you. Okay. So is that what does that mean exactly? Are you saying it stays consistent? Then what is it? Are you sure? That is the independent variable, what you're changing in the experiment.
Temperature that stays the same.
The easiest way you can actually describe the dependent variable is what you are measuring. What you are measuring in the experiment. What is it that you are using as your data? Okay. If you're collecting data in an experiment, that will be your dependent variable. If you are changing something in the experiment, that is your independent variable, right? So here, what is being changed and what is being measured?
Doesn't change.
No, dependent is not changing. That is your. The reason why, it's actually in the name itself, right? The dependent variable is depending on the independent variable because your data can be influenced by what you change, right? That's why it's depending on that variable, right? So just associate that idea, right? Dependent variable is depending on what you're changing. That's why it's depending on your independent variables. Okay, I can change. Yeah, I'm not going to give the answers. What are the answers? Can I get Gavin? Okay, this one's not related to cellular as actually it is, but you can still find out the answers. What are you changing in this experiment? Independent variable. The oxygen. Are you sure? Read the question, guys. Always read the question. You will be surprised how often they give away that answer in the question. First sentence itself, right? Matthew investigated how changes in environmental temperature affected oxygen and carbon dioxide levels. Okay, that is almost like an aim if you actually look at it, right? Investigated how changes in the. So in your question, you'll be shocked how often they include your aim without even realizing it, right? You need to be able to extract the variables from the aim that they've been given, that they are giving. Okay, so what is changing here? Among the, so you'd be, if you just answered temperature, immediately wrong. Okay, you need to be specific. Okay, you can't just say temperature of what? The environmental temperature, that's good enough already. Okay, you need to be somewhat specific. So either you say environmental temperature, temperature inside the enclosure, it doesn't matter, but you need to be somewhat specific. Okay, writing one word answers for your variables is not something you're going to get marks for. Okay, dependent variable, what is it? Yeah, levels. Yeah, that's spot on, right? Oxygen and carbon dioxide levels. You could write around the cockroach, but that is not important. Not really too important. So, like I said, temperature of the chamber or environmental temperature. Okay, if you wrote temperature or heat lamp, like I said, not giving any marks for that. In science, you guys need to be somewhat specific, indicating temperature of what? Because when you say something as vague as temperature, I can say, oh, do you mean the temperature of outside the cage? Why do I need to change that? You need to say it's inside the cage or the environmental temperature in general. All right, this one's a bit more involved. This is a continuation of the previous question, by the way. So this one is saying that it's measuring the carbon dioxide levels and CO2 levels, sorry, the CO2 levels and the oxygen levels with different temperatures. So these three graphs are showing that, right? So these two are showing your carbon dioxide and oxygen. This is showing your temperature. So what, using the graphical data, describe the changes in the levels of carbon dioxide and oxygen when the temperature in the chamber was kept constant compared to when the temperature is decreasing. So in a complicated question like this, you guys really need to understand what the question is asking. Okay, first, note down, describe changes in CO2 and oxygen levels, right? That is your independent variable. Sorry, that is your dependent variables, right? They want you to describe your dependent variables, and then they've also given when the temperature in the chamber is kept constant compared to when it's decreasing. So let's look at your temperature graph, right? When we look at your temperature graph, there are two trends that you can see. One trend is when the temperature is kept constant, and another trend when the temperature is decreasing. They basically want you to use the dependent variables and make a statement of what was the uh, dependent variables like when the temperature was kept constant, and then what were the dependent variables like when the temperature is decreasing. So you'd have to analyze all the graphs that are present to make that assumption. So if you had to describe the dependent variables while the temperature is kept constant versus the dependent variables when the temperature is decreasing, how would you make that contrast? So it's important that you're contrasting them, meaning that you are differentiating them, saying that when the temperature is kept constant, what are the CO2 and oxygen levels doing? When the temperature is decreasing, what are the levels doing? You need to make them different from each other. Okay, that's what it means to compare and contrast. So when the temperature is kept constant, what is happening to the CO2 and oxygen levels? Who can tell?
Carbon dioxide is increasing the oxygen.
See, that is just stating one thing. You are just saying increasing and decreasing. What about when the temperature is decreasing? Isn't that the same thing that's happening? They're both when it's constant and when it's changing, both are going in an increasing trend. So just saying increasing is not good enough because both are still increasing. You need to go more specific than that. So saying that when the temperature is kept constant, the rate at which, so you would say that the carbon dioxide levels are increasing at a faster rate compared to when the temperature is decreasing, because the rate at which it's increasing is different, right? When the temperature is kept constant, the rate at which it's increasing is higher compared to when the temperature is decreasing, the rate is slightly lesser. So you need to make that differentiation when you're giving your answer. Same thing with your oxygen levels. When the temperature is kept constant, the rate at which the oxygen levels are decreasing is much higher compared to when the temperature is decreasing, where the rate is decreasing at a slower rate. Okay, so you can describe it in different ways depending on how you want to write the answer, but essentially you need to make that contrast. So when this temperature is constant, the levels of CO2 increase sharply and slowly rise with decreasing temperature. So that's the most easiest way you can write it down. You can use different ways to describe it, but one thing to consider is how many marks is the question worth? How long do you want to write a response? Do not waste time writing a long response for a 2-mark question. That just makes it so that you don't have time for other questions later on. So make your response concise and to the point. Okay. Understand what the question is asking. Make sure you satisfy it within the context of 2 marks. Okay. Now the second one, and the levels of oxygen decrease sharply and then slowly decrease with the decreasing temperature. Any questions? So you can see that a lot of people so when you look at the reporting, people either were really aware of how to answer these kind of questions, how to contrast accurately, or people really didn't know how to do it. A very big disparity. There is no middle ground. Okay, a lot of people either knew how to answer or don't know how to answer. I hope you guys know how to answer now, since you have gone through it.